Hey, hello, my name is McCabe, and I'm going to be presenting on a project on the impact of estrogen treatment on prostate cancer and what implications this might have for transgender healthcare. Now, prostate cancer is a prominent area of research, and one aspect of that is the impact that estrogen has on prostate cancer. This is not exactly clear because there's two different types of estrogen receptor, alpha and beta, which seem to have opposite proliferating and anti-proliferating impact on cancerous effects. So one area where there's a particular interest is transgender healthcare, where prostates are very rarely removed during transition, but levels of estrogen are highly elevated, and there's very little research in this area, so this study aimed to investigate that. The first part of that was to investigate the presence of the receptors as this varies between cell lines. We're using PC3s here, and we used an immunofluorescence as well as a PCR in order to investigate the receptor expression with MCF7 and MDA MB231 breast cancer cells as positive and negative controls. The immunofluorescence test had inconclusive results because the negative controls showed fluorescence, but the PCR did show an amplicon at the correct size on the agarose gel for both the alpha and the beta receptors, which indicates that they are both present. Now, with this in mind, we moved on to do 3 types of assays to test the different ways this might affect prostate cancer. These included a clonogenic assay to test cell proliferation, an MTT assay to test cell viability, and a scratch assay to test cell migration. All of them used ethanol as a vehicle because estradiol does not dissolve well in things like PBS. There was a significant impact on the ability of cells to proliferate when treated with estrogen regardless of treatment. And the same was seen for cell viability, where there was a decrease across all of the tested treatments compared to ethanol-treated cells. Cell migration, however, was not impacted in any significant way, which may indicate that the impact of estrogen is more complicated, and it depends on the factor. The big takeaway, however, is that none of these have indicated an increase in cancerous effects that could classify prostate cancer as a significant risk factor when someone is being considered for hormone therapy with estrogen. This would also seem to indicate that estrogen receptor beta has a larger impact, but that is not directly studied here. This fits as well with the low incidence of prostate cancer in the transgender population. There are a number of possible future directions for this area of study, including testing androgen deprivation, non-cancerous prostate cells that may have different expression of estrogen receptors. And I hope to see more interest in this field in the future.